Good morning. I'm going to get started on time because I want to use every minute of our symposium time to talk about a topic that is not only fascinating, but is critically important because small molecules, while they have been the bedrock of neurotherapeutics, will be also joined by neuromodulation. And at this meeting, the first time I came to this meeting was about a decade ago. There were no devices. Every year there have been a few more, and now it's really become a key part of our discussion and also a real focus of exciting translational research. So if I can have there we go. So today we're talking about direct brain stimulation. There are many ways to apply energy to the brain, and some of them are non-invasive, some are involved with stimulation of the peripheral nerve, and those topics will be discussed in other symposium during this meeting, but today we're talking about direct brain stimulation. So from my simplistic way of looking at it, there's two topics or two areas that we need to figure out within the field. And the first is, how should we be delivering stimulation? Should we be delivering stimulation in a continuous or scheduled fashion? In other words, open loop? Or should we be providing stimulation in a responsive way, meaning that stimulation is only provided in response to a certain event? That might be electrophysiologically, it might be temperature, it might be neurotransmitters, but it's triggered. Another pressing need is to understand where we should be providing stimulation. What are the targets? Is the target a focus, a subcortical focus, deep brain, thalamic, or is it in the cortex, or is it in networks? What are the applications for these therapies? Because I'll just begin by saying that all of them work. All of them work, and there are many applications that we have excellent Class 1 data demonstrating safety and effectiveness. One approved therapy is open loop stimulation of the thalamus, also known as DBS. And there are a number of approved indications for use, including Parkinson's disease, probably what we're all most familiar with, a long and important history, a profound breakthrough in the treatment of movement disorders, as well as essential tremor, dystonia, OCD, and then recently epilepsy. This approach is most commonly stimulation of a specific nucleus of the thalamus, and that nucleus varies depending on the disorder that is being treated. Another approved deep brain or direct brain stimulation is a closed loop system, which delivers stimulation to a pre-specified target or target that is individualized to the patient. In this situation, there is an active sensing and then a response to an event with stimulation, a different approach shown to be safe and effective for partial onset epilepsy. There are also a number of active investigations in the area of brain stimulation. A number of investigators who are looking at motor cortex stimulation for pain, others who are looking at cerebellar stimulation for stroke rehabilitation, and then a great deal of interest in affective disorders, psychiatric disorders, including, of course, depression. There were 2 failed trials of stimulation for depression in the recent past, but that doesn't mean it's not going to work. I think everybody is quite optimistic that it will work, and here's just one illustration of putative targets for neurostimulation to treat depression. DARPA has put a lot of its money and also its reputation in the area of neuromodulation, direct brain stimulation, and that includes in neuropsychiatric disorders and in restoring active memory in people who have experienced brain trauma or for any other condition. So today we're going to begin with Dr. John Rolston, who is a functional neurosurgeon at University of Utah, and with his group there has done some really, I think, incredible work on defining the modulation or the networks that should be modulated for specific brain disorders. That will be followed by Ken Baker from Cleveland Clinic, who is part of the group that's doing fascinating work funded through the Brain Initiative in post-stroke motor rehabilitation. Steve Goetz from Medtronic, who is obviously very involved in thinking about the future of DBS, and I heard him speak recently, and his kind of overarching view of how to apply this I thought was really intriguing, and he was talking about treating symptoms and not necessarily treating a specific disease, because we're treating networks. And then it's fabulous because Carlos Peña, who is running the group at Centers for Devices in Radiological Health that is involved in regulating devices for rehabilitation and neurological disorders, has joined us and will talk to us about how his division is thinking about regulation of this explosion in medical devices. So I will begin by turning it over to, that's my last slide, so we'll switch to Dr. Rolston's slide. To Dr. Rolston.
All right, so thank you for the invitation to speak here. So I'm John Rolston, and I'm the Director of Functional Neurosurgery at the University of Utah, and also a member of the Scientific Computing and Imaging Institute there. And today I'll be talking about how networks are more important for neuromodulation therapies than we might have been giving them credit for. So first, my disclosures. So funding from the NIH, as well as the CNS, Epilepsy Research Foundation, Coulter Foundation, USTAR, stock options in Axion Biosystems, which makes multi-electrode arrays for in vivo research and in vitro research primarily, and then consulting agreements with both NeuroPace and Medtronic. So I'm going to start with a commercial from Canada in 1991. Can we get the sound up for this?
Toast is burning.
Toast is not.
This was forced viewing in Canada in the 90s.
Every time she has a seizure, she smells something burning. Now, if we can provoke that smell by probing the surface of the brain, we'll find the source of the seizures. Mrs. Gold, do you feel anything?
I can see the most wonderful lights.
And now what do you feel?
Did you pour cold water on my hand, Dr. Penfield?
Now what?
Oh,
What is it, Mrs. Gold?
burnt toast. Dr. Penfield, I can smell burnt toast. Dr. Wilder Penfield. He cured my seizures and hundreds more. They say he drew the roadmap of the human brain. We just called him the greatest Canadian alive.
So it's comical, but interesting. The way that they did this brain mapping really was started in humans in the 19-teens. Cushing was the first one to do it in humans for brain tumor mapping. So he would use a bipolar probe to deliver electrical stimulation to the surface of the brain and either provoke activity, so evoke movements in the patient, or suppress activity, things like speech arrest. And that was used to infer the activity or the responsibility of that brain area right beneath the electrode. They initially used 60 hertz stimulation in the United States and 50 hertz in Europe, primarily because the early systems they used just coupled into the wall and used the AC and rectified it to make a sort of an AC pass. So they still use, and I guess is there a laser pointer? There it is. So the system we use right now is this E.D.E.N stimulator right here, which kind of looks like a box from Radio Shack in the 70s. This is what we still use today in the operating room. It's still the same bipolar probe and still uses 60 hertz stimulation. There's been very little investigation of how these parameters could be improved over time. And this is an article from just a couple years ago, just showing this sort of the mentality that we use for brain mapping, where we stimulate, put little ticker tapes down where we think important functions are, and then resect areas that we think are not involved in those functions. And we use the same mentality for epilepsy surgery too, where we try to find where the seizure focus is, where we believe the seizures are coming from, and try to pinpoint one precise localized area, usually with a grid of electrodes, to take out. But that's a very phrenological view of the brain. So thinking about that there's a particular spot in the brain that is responsible for speech, or a particular spot that's responsible for a particular movement, is this idea of this functional segregation, or there's specific areas that are responsible for a single function. This is opposed to thinking of an integrated view of the brain or a connective view of the brain. And this is a kind of an issue with our field, is that we still, even recently, put a lot more faith in this view of the brain, the sort of idea that there are particular spots that do particular functions. And there's much more publications than that, the idea that there's localized brain function rather than the idea that there's network responsibilities for these functions. And that gets us into problems occasionally. So why should we think about networks? So I think the best example is the lobster, so bear with me. This will have bearing on humans. Eve Marder of Brandeis did a lot of really sort of pivotal investigations in the stomatogastric ganglion of the lobster to figure out how those neurons and how that digestive system in the lobster worked. If you patch onto a single cell during the sort of the peristaltic type movements, you'll see these bursts of action potentials that go on. However, if you cut the connections and leave that neuron just by itself, you don't see any activity like this. So the bursting activity that we see, the coordinated activity from all this network of neurons, only emerges when the neurons are in this network. So you would never be able to find this behavior just by looking at cells in isolation. You need the network to see that behavior. It's also a little more complicated because if you look at the sort of parameters for each neuron, they can vary wildly to give you the same sort of behavior. So here's two model neural networks that she created. They have pretty much identical behavior phenomenologically, so I couldn't tell the difference between this network on the left and this network on the right. But when you look at the actual parameters that are embedded in these networks, there's differences in conductivities at synapses that can vary by an order of 10 or more, and you still get the identical function. So the nodes by themselves aren't necessarily the same in between two networks that can give you the same behavior. So you can imagine how if you try to take this to a human level, that you might find nodes in a particular patient that are responsible for something like speech, but in a different patient might be responsible in a totally different way or part of a totally different network. So this is work we did at UCSF looking at speech mapping in particular. So here we had patients who were outside the operating room. They had high-density electrocorticography grids on their cortex, and we would stimulate patiently at the bedside to see which of these electrodes we could induce speech arrest or changes in speech function at. Here's sort of two examples. So this little blue electrode right here caused speech arrest when you stimulated it, and this one, which is about 8 millimeters away, did not cause any speech changes. And the main difference with this was not the anatomy of the location, but really the connectivity of the location. So we used imaginary coherence as a surrogate marker for functional connectivity and looked to see where these areas were most strongly connected to. And the one that produced this speech arrest was really strongly connected to the posterior temporal lobe, where the one that didn't cause any problems wasn't. So again, it's not necessarily the area itself, it's the network that's causing these changes. We did the same thing looking at somatosensation in epilepsy patients as well. So again, we did this little bipolar stimulation in a part of the sensory cortex. And what we did was we found a sensory threshold where we could stimulate at a certain current intensity, let's say 2.5 milliamps or so, and half the time the patient would report a sensation and half the time they wouldn't. So we would give an identical stimulus, and the only difference would be the patient's perception of the stimulus and not the actual stimulus itself. And what we saw is when the patient perceived something, a huge network was sort of activated across the span of the cortical gyri, across the cortex, including both sensory and motor cortices. And you see this predominantly in the high gamma band. This is a little different than natural touch, which is much more circumscribed. But the main difference here is if you looked at the ones that are non-perceived, there's not a good figure for this, unfortunately, the evoked activity there is really circumscribed, very small amount, but when it's perceived, you have a large network that's activated. So again, it's network effects that are responsible for what we're perceiving in these patients and what we're doing in these patients. So has this network sort of theorizing been useful for treating patients? Yes, of course. So I think the best example is deep brain stimulation for movement disorders. So we have this canonical circuit of the basal ganglia, which was delineated by Malin DeLong and others many years ago, predominantly in MPTP monkeys. And they found that there was hyperactivity often in the globus pallidus interna, which was suppressing the thalamus and therefore inducing what they thought was bradykinesia. So that's the kind of the high-level circuit diagram they made up. And so based upon this, we thought that if you just cut out the globus pallidus, you can cure these patients or help with their movement disorder. And that's what we did for a long time in the 60s and 70s and 80s. We did pallidotomies and subthalamotomies. We eventually transitioned to deep brain stimulation on the idea that doing stimulation was essentially the same as doing a lesion. So putting an electrode and turning it on would shut off that area of the brain. It's probably much more complicated. It definitely is much more complicated than that. And a lot of our initial theorizing doesn't really pan out when you look at what the neurons are actually doing, especially when you sort of segregate the tissue optogenetically. But the idea of using this network and intervening in a particular node in the network was really the most effective way to treat this disease. People had tried attacking this node of the network, the motor cortex, and just basically extirpating the motor cortex, which would also cure the tremor, but left the people hemiplegic. Another idea of using the network is, as Martha mentioned, the anterior nucleus of the thalamus stimulation with DBS. So in the circuit of Papes, the anterior nucleus of the thalamus is an integrated node here that's responsible for sort of the reentering activity from the limbic system. So again, the hypothesis was if you can silence this area, the anterior nucleus, you can disrupt that reentering activity and suppress seizures, which appears to be the case. Although again, once we learn more about what's actually going on and what's actually happening with the stimulation, it's probably going to be much more complicated than that. Depression, which we also alluded to, came along slightly different ideas. When Helen Mayberg was working on this, she initially was doing PET scans in patients. So this is a PET scan activation in responders to SSRIs with depression and non-responders. And one thing they noticed was this area right here, the subgenual cingulate, area 25, Broman areas 25, had decreased activity in the responders to SSRIs and not in the non-responders. So she went to Andres Lozano at the University of Toronto and said, how can we do this in patients? How can we make this area less active? And the idea again was, well, let's use deep brain stimulation since that seems to be a sort of electrical lesion. We'll put electrodes in there, sort of turn this part off and try to cure the depression. They did that in a few patients, a handful of patients, published a Neuron paper about it, and that sort of spurred, because of its success, a large clinical trial, the Broaden clinical trial. That trial ultimately failed. But we learned a lot by doing these studies. And what we found was that it's probably not that particular area of the brain, but the network that you activate when you stimulate that is responsible for success. So when they looked at the responders and they did computational modeling to see what areas of the brain were activated at effective stimulation parameters, the effective stimulation parameters activated a large swath of the sort of orbitofrontal areas in these limbic networks, whereas the ineffective stimulation didn't. So again, it's not this particular single node, the single focus, it's a network effect that's successful for treating a lot of these diseases. Further evidence that networks are really important, there's a recent paper out of Matt Fox's group looking at network imaging to sort of infer functions or infer pathology for different diseases. Here they looked at individual modalities to try to find aberrations in particular pathologies. This example is for Alzheimer's. So looking at an MRI, functional connectivity networks to see what was sort of aberrantly active or inactive in Alzheimer's, they found mesial temporal structures. In PET scans, they found occipital structures. In SPECT scans, they found, again, mesial temporal. When you combine all these across modalities, you find nothing sort of survives the meta-analysis. So you get really an uninformative result here. Whereas if you take these activated areas or inactivated areas and use them as seeds to look at the network that's involved, so looking at what network you're localizing rather than just what individual node in that network, and then when you look across these different modalities and then combine them, you get these preserved networks. So what they're finding is that an individual modality like MRI, PET, or SPECT might find just one of these nodes in these really involved heterogeneous networks. But when you look at the network actually, not just the node that's abnormal, then you find what's sort of the crux of the matter for these patients. What gets a little more dicey with all these ideas of networks is that networks change over time. They're very dynamic. A great example of this over the idea of days to weeks is a paper by Danny Bassett at U.Penn. So this was looking at fMRI during a visual motor learning task. So the patient would learn a response based upon a visual cue. And you can see that there's sort of this new connectivity that evolves when the patient learns this game. So there's a lot of connectivity between the visual and motor areas, but also these higher, like yellower colors are higher connectivity. There's a lot of connections between areas that are kind of extraneous. When the patient starts to learn this task and gets better and better at it, all these kind of light areas disappear, so the brain becomes very efficient. So that connectivity, the weights that we see from these different modalities are not static over the sort of days to weeks type timescales. When we look at even shorter timescales that we can get, for instance, from electrocorticography, that same truth holds. So you can take a electrocorticogram and make these little sliding windows and compute the connectivity matrix over each of these time points, and that changes over time. So the network configuration is changing at probably the second or even longer time scale. So there's a lot of dynamics on these rapid time scales too that are changing as these patients evolve through seizures or interictal activity. This is recent data from our lab at the University of Utah. So we unfortunately inherited a large trove of Utah array data beginning at Utah. So these are these small 4x4 millimeter electrode arrays. Each one is able to record from single units. And here we are sort of mining data from patients that had previously been implanted with these. These are human epilepsy patients, and looking at things like interictal spikes. So when you look at an interictal spike across this array of 96 electrodes, they look kind of synchronous. But when you start to zoom into these, you find that there's actually slight delays for the interictal spike upon each electrode. And you can fit a 2D plane in there, do a linear regression, and actually see that there's a propagation of this wave. So the interictal spikes are actually traveling waves through the brain. They're not just these isolated single events. And when you look at these, for each individual interictal spike, you kind of plot the direction they're propagating and do a histogram. You see that there's a definitely consistent direction, which actually tends to be opposite of the ictal wavefront. And so the point of bringing this up is that even at these sub-millisecond timescales, there's dynamics in all this activity that you wouldn't capture if you're looking at either static stuff at resting state or these even slow speeds at the regular ECOG resolution. So how do we put this into practice? How do we take these lessons we're learning and actually do something to modulate these networks? So the sort of workflow that a lot of us are using right now is to first choose some sort of modality to figure out what the nodes that you're looking at and what the edges you're going to be looking at are to define these networks. So you can do something like MRI, DTI, MEG, some way to get a sort of representation of the brain. Then you need to define what regions you're interested in. So there's many atlases out there. There's the MNI atlas, the AAL2, the Brainatome. All these different atlases have different ways of divvying up the surface of the brain and are all very different than the original sort of Brodmann areas that people are used to. Once you find your preferred way to break up the brain into different regions, you have to define edges between these nodes. So that can be with something like electrocorticography or fMRI weightings, basically a way to say that how two different regions are connected. Then you have these connectivity matrices. You can binarize them or threshold them to find sort of simpler representations of these matrices. And once you get that matrix, that's when you start doing the fun stuff of graph theory. So you can apply the tools of computer science and graph theory that have been around for hundreds of years to these brain networks that you're developing. And that brings us to control theory. So control theory is a very healthy engineering way of looking at these networks. There's been a lot of sort of fresh new wave of interest in this in the past decade. The idea here is that if you have some sort of graph that you've generated from your non-invasive imaging or your invasive electrocorticography, you can look at particular states of that network, and you can see what kind of states you might want to get to, for instance. So if you have a state that you think is epileptogenic and one that's non-epileptogenic, then you can do basically a Hamiltonian on this and sort of do the mathematics and figure out what nodes are the most effective to change that state for you. So there's this whole engineering theory about using control nodes to change the state of the network. So this would tell you that maybe it's not the most active node, for instance, in a seizure. Maybe it's not the epileptic focus that's the one that's most effective in changing the behavior. Maybe there's a different node that you should be modulating. That's not the one that's just the most obvious one because of its activity. People have applied this to other realms. So here's looking at just a regular control subjects and looking at what areas are most controllable versus least controllable. So the ones that are easy to control are the ones that are involved in these default mode networks. That's the red areas right here. The ones that are hard to control or useful for controlling hard to reach states tend to be the sort of cognitive control functions, the prefrontal cortices. They've looked at using this for TBI patients. So in TBI patients, they found that there is this disruption in the way the controllability appears when you do these analyses. In the TBI patients, it's harder for them to move to the sort of hard-to-reach areas than it is for a control subject. So it's harder for them to get that healthy state, and that's an indication of why they might have these cognitive symptoms that they're plagued by. So once you identify what areas you might want to stimulate, what are the control nodes you want to get to, how would you actually deliver a device to do that? And that's where we get into modeling. So this is the work that we're doing now with Chris Butson at the Ski Institute at the University of Utah. And this is a stereo EEG finite element model of an electrode in one of my patients. And here we're seeing what sort of activation you get in fibers of passage. So these are axons that are moving by the stereo EEG electrode. And this is the volume of tissue that would be activated. This is simulated by stimulating these particular two electrodes in a bipolar montage at a certain current intensity. And you can see that this pathway is activated, and you can use that to predict the effects we see, which are if we stimulate over here, for instance, we see big responses a few centimeters away. And we can predict that by how the areas are connected by their tractography. So this is the sort of missing link, is you want to get an idea of which nodes are important by using some sort of network analysis or network imaging or even electrocorticography, and then trying to figure out how to best influence those nodes. Then you use computational modeling to see what you're stimulating. This is that same sort of methodology applied to a patient with a NeuroPace device. Here is the amygdala sort of lit up in this kind of purplish, and then the red is the hippocampus. And here are the volumes of tissue that are activated at a particular stimulation amplitude with these different electrode contacts. So now you can say, oh, I have this electrode in. I want to know the best way to modulate a particular network, the best way to control it. You can kind of work backward to see what parameters you would need to use. And the reason this is so powerful is not only that we're currently placing electrodes kind of in a trial and error way. We suppose we want to get to the epileptic focus or an area we think is involved in the network. We stick the electrode in and turn it on and spend a few months trying to program the best parameters. If we can use the engineering tools that we have from other domains, we can prospectively do this. We can do this in advance and say, well, if I want to activate a particular network, here's the location I would do that in, here's the parameters I would use to do that. To take that entire, many year time, sometimes process of programming and make it an instantaneous preoperative type planning procedure. So with that, I'll just give thanks to all my colleagues at UCSF, the University of Utah, as well as Columbia and now IMC. And you can do it questions now or questions at the end? We're going to do after. Okay, perfect. All right.
Thank you so much, and please jot your questions down because we're going to have a discussion, and I'm going to turn it over to Ken to talk about stimulation in the cerebellum.
Thank you, and thank you for the invitation to come and speak here today. My name is Ken Baker. I'm from the Neurosciences Department at the Cleveland Clinic Lerner Research Institute, and I'll be talking about a fairly specific project that I've been working on with Andre Machado at the Cleveland Clinic, representing essentially about 15 years worth of research. I don't know what I'm doing. There we go. Some disclosures and research support. Gratitude. I do not have a direct conflict of interest with the sponsor of this trial, though I do work for the company that spun it off, so however that works. Essentially, I'll be talking today about the E.D.E.N trial, which is our effort to enhance motor rehabilitation for chronic post-stroke patients. I'm just going to go over what we consider to be the need and the rationale, our hypothesis, experimental approach, the preclinical evidence that we think supports this trial, and then a little bit about the first in-human study that started 2 years ago. So stroke numbers, it's obviously a leading cause of long-term disability, both in the United States as well as worldwide. About 800,000 new cases per year, 17 million new cases per year estimated worldwide, which comes down to about 1 every 2 seconds. At least 50% of those survivors are disabled at 6 months post-stroke, and this in the US alone translates to about $37 billion in indirect and direct cost annually. Hyperacute and acute care has greatly improved over the past 2 decades. And the consequences of that are that more patients are surviving strokes and more patients are surviving more severe strokes, which leads again to the work that we're doing. And finally, that spontaneous biological recovery, standard of care, state-of-the-art care for these patients is still physical therapy, but it's inherently limited. So our goal, like the goals of many others, contemporarily and in the past, has been to try to find ways to extend or enhance the period of motor recovery in these patients. This has been tried, as was mentioned during the introduction, through non-invasive means with transcranial magnetic stimulation, direct current stimulation, peripheral nerve stimulation, as well as more recently invasive cortical epidural stimulation trials, the North Star trial being one of those, which failed to meet its endpoints in human trials, and finally more recently vagal nerve stimulation. So tried in many forms, but the results to date have either been modest or highly inconsistent. Building upon the last talk, Andre and I, essentially 15 years ago, were looking for ways that we could enter this network to try to do the same thing that many of those studies were doing, which was to try to enhance cortical excitability and enhance the functional reorganization. Our choice of the pathway that we thought might be a way to chronically modulate this activity was to enter at the level of the dentate nucleus, which represents the starting point of a disynaptic net excitatory pathway that connects the output. It represents essentially the ascending output of the cerebellum, and it connects the cerebellum via multiple thalamic subnuclei to widespread cortical regions. So it's not that we thought there was a pathology in this network, and the pathology in the dentate. It gave us a focused, efficient nodal point to try to activate a pathway that we thought could have widespread, robust effects on cerebrocortical activity. Certainly at the time that this 2004 drawing that went into our first R21 effort, didn't go well, was made, we certainly knew at that point that single pulse stimulation in anything ranging from rats to apes, single pulse stimulation of the dentate would result about 3 to 5 milliseconds later in a robust increase in neuronal activity in the M1 cortex, the primary motor cortex. We also knew from the work of Dumm and Strick, who did beautiful track tracing studies as well as other imaging studies, that unlike what we drew here on our first drawing, the connections didn't just cover the motor cortex, but actually spread all the way to the frontal pole as well as posterior parietal regions. So we thought we had a pathway that would allow us to, again, modulate cerebrocortical activity across robust areas of the brain, or widespread areas of the brain, certainly areas that would represent the perilesional cortex, where we were trying to modulate to promote functional reorganization. So we asked ourselves, if the single pulse stimulation can increase cortical activity, would we be able to do that chronically? So can we use chronic electrical stimulation of this endogenous ascending cerebello-cortical pathway to enhance chronic post-stroke motor rehabilitation? Our hypothesis, like those of others, those also involved in neurostimulation, was to try to increase perilesional excitability, which is coupled with neurogenesis, increase in growth factors, in order to promote plasticity. We're not looking here, and I'm not talking here about an acute therapy or even a subacute therapy at this point in its current form, and we're not trying to affect the stroke core, and it's not also a prosthesis or a substitution. Rather, I'm trying to work through an endogenous pathway to reestablish a window or a physiological environment that is more conducive to functional reorganization. So our work in the preclinical ischemia model, we used the Long-Evans rodent for most of our work, and a couple different stroke models. The dentate nucleus in the rodent is called the lateral cerebellar nucleus, so we had to implant electrodes in that nucleus. And the first thing we wanted to find out was whether or not we could have a more prolonged effect on cerebrocortical excitability through stimulation of that structure and that pathway. The setup shown here is intracortical microstimulation, evoked motor evoked potentials, essentially stimulating the cortex and watching the forelimb or the hind limb twitch. And we would pair that with and without stimulation, concurrent stimulation of the dentate nucleus. So 3-pulse stimulation of the motor cortex evokes a motor evoked potential, and we do off versus on stimulation. did a block paradigm shown here, where off and a certain particular frequency of DBS is done over alternating 10-minute intervals. So motor evoked potential is induced periodically throughout the 10 minutes of the off period. We turn stimulation on, turn it back off, turn it on again and off again. And what we found is that we could modulate cortical excitability, but that modulation is frequency sensitive. So here, showing the average across multiple animals, showing the 10 minutes of off period averaged and the data normalized to that off period, we found that 20 hertz stimulation of the dentate did increase our motor evoked potential, but that 30 and 40 did it better. And once we got to 50 hertz stimulation, we did get an initial acute enhancement of response, followed by, however, after a few minutes, a decrement in response, and that higher frequency stimulation, those used for Parkinson's disease and essential tremor, actually caused a trend towards reduced cortical excitability. That same frequency of stimulation was applied for behavioral effects in different models of ischemic stroke, showing here the endothelin-1 model that we went to after the 3-vessel occlusion model. Animals are pre-trained on a certain motor task, in this case, reaching for pasta through a narrow slit outside of their home cage. A lesion is created over their primary motor cortex. We can quantify and characterize that lesion. And the animals are then separated into groups that are going to receive stimulation shown in red or not receive stimulation shown in blue. Animals are pre-trained to plateau. The stroke is induced, which results in a behavioral decrement. Stimulation is started several weeks later, and we look for this separation, which seems to suggest to us that we're getting a benefit from DBS. We wanted to confirm that, consistent with our hypothesis, that this might actually be related to functional perilesional reorganization, so we compared again with ICMS, the naive versus the sham-treated animals, and in this case just showing two different treatment groups, the motor representation across the cortex of the rat. In the naive animal, we have significant representation for the distal and proximal forelimb. In these sham-treated animals who received an electrode but no stimulation, that representation is lost, but we seem to see an increase in that representation following therapy in our treated animals. Consistent with that, we went steps further to try to look at the area around our lesion, our perilesional cortex, as it were, using 3D electron microscopy methods to count the number of synapses, and indeed showing here in gray the sham-treated animals versus the blue for the DBS-treated animals in samples 1, 2, and 3, the average number of synapses per sample, which again was further supported by an increase in markers within the perilesional area of synaptophysin and other markers that suggest long-term potentiation. And that essentially led us, after about 12 years, to human translation. So the trial is called E.D.E.N, and again, it's for dentate nucleus deep brain stimulation to enhance chronic post-stroke motor rehab. At this point, it's a phase 1 safety and feasibility trial looking to enroll 12 patients. Those patients will have had a unilateral stroke in the territory of the middle cerebral artery 12 to 36 months prior, and they will have persistent moderate to severe residual unilateral upper extremity hemiparesis. Right now, our Fugl-Meyer cutoff is a 33. The upper extremity scale has a range up to 66. Just to note, the patients, I'm going to show you the first 6 patients really today. Those patients had a score of less than 20, as that was the initial requirement from the IDE, which only more recently extended up to 33. So our initial 6 patients in the study were essentially in the bottom third of that assessment scale. But we did want to make sure that there was reasonable pathways still intact in the corticospinal pathways, and we used TMS as a notable inclusion criteria to make sure that we could elicit motor evoked potential. The design, very briefly, is a sort of a lopsided wedding cake. We start with the baseline prior to implant. The patients undergo about 2 months worth of rehabilitation. And that's really there just to ensure that they don't show additional recovery from rehabilitation alone, that maybe they were underserved, maybe there are still mechanisms before going on to an invasive procedure. Assuming that they don't show significant change across that 2 months, they undergo an implant with rehabilitation stalled for about a month for recovery, after which rehabilitation starts again for a period of 2 months before we start to titrate and program. That programming takes about a month. We go through various settings and metrics that I'll touch on at the very end here to try to figure out what biomarker we might find that suggests that we are having an effect, the effect that we want. After that, after the setting is determined, the patients go through an experimental phase of about 4 months of stimulation, now up to 8 months for reasons I'll show in a moment, at which point stimulation is turned off. Rehabilitation continues for an additional 2 months to ensure that any gains that might have been made are sustained before the device is explanted and the patient moves to long-term follow-up. So a number of metrics. It's a phase 1 trial, so obviously safety and feasibility is first and foremost. As I mentioned, the Fugl-Meyer upper extremity assessment is our primary outcome metric. We're looking at medical history, we're looking at different functional imaging techniques and electrophysiology in the hopes of helping to better characterize the patients and the variability that I'll show you in just a moment, and make better decisions about designing future trials. The surgical approach uses standard deep brain stimulation hardware. There's a Lexel frame shown secured to a patient's head, but in this case, in contrast to most DBS procedures that you'll see, the patient is anesthetized and belly down because our approach goes through the suboccipital regions through the posterior fossa. So all of the targeting is imaging guided, and here's the surgeon in the patient's head with an O-arm to help guide the trajectory. The end result, shown in the upper left, is a pacemaker-like device implanted in the chest with a wire coming up to an extension and a connection at the level of the skull. The excess lead, because it's one size fits all, is tucked under the muscle against the skull, and you can see the tip of the lead entering through a burr hole in the posterior fossa about 4 centimeters into the cerebellum. You can see that a little better on the right. The cranial defect is here. This is the wire now entering the dentate nucleus. The hyperintense, when it got brighter, represented the actual individual contacts of the DBS lead, spanning the length of the dentate nucleus. So our current status, we have 6 patients implanted. 2 have fully completed the trial. 3 are in the DBS plus rehab, essentially the experimental treatment phase, and 1 patient is still being programmed. And there have been no serious adverse events from this. So as I said, our primary efficacy is the Fugl-Meyer upper extremity with a max score of 66. This essentially follows along the same experimental design I showed you earlier. I mentioned all the patients that we started with had Fugl-Meyers of less than 20. So prior to implant, they had 2 scores representing the 2 months of their physical therapy, re-entering physical therapy with DBS turned on at about month 4. Looking across the experimental phase, I mentioned earlier that it was originally a 4-month design. It went to an 8-month design, and that was E.D.E.N.'s fault, shown up here. We got to the 4th month, and she was still showing month-to-month improvements. So we went back to the FDA and asked and changed our algorithm to do more of a moving average to decide when to discontinue therapy for the patients. E.D.E.N. 2 and 3 shown here basically plateaued fairly quickly and only went through 4 months of therapy. So showing that in a different format, normalized essentially to the final pre-treatment assessment, E.D.E.N. was fortunately and unfortunately a stellar responder, but everything else after that, essentially the next 2 patients have shown relatively modest improvements. The 3.5 was designated in the North Star trial as a clinically meaningful outcome. Obviously, that needs to be weighted against the cost and invasiveness and risks of the procedure, but it does provide a precedent for what we're looking at. So that variability is something that we're going to have to obviously explain as we continue to move forward. And unlike, again, the cases of essential tremor and Parkinson's disease, which do have heterogeneity, These are the first 5 entrants into the study. I realize it's only a single coronal section, but it helps to simplify things. And still denotes that we have a high degree of variability in the morphology, the location, the volume of the lesion, despite fairly similar scores on the Fugl-Meyer assessment. So the type of variability that we're seeing both in the imaging, the type of variability we're seeing in the outcomes is okay, so long as we're doing what we can to try to learn from it. And that is one of the things that the Brain Initiative Award did allow us to extend the trial beyond its original design to gather resting state, type imaging, PET imaging, and a host of electrophysiology measures to try to better understand this variability. So we need to answer questions related to patient selection, surgical targeting. Like most targets we have for deep brain stimulation, there are motor and non-motor areas of the dentate, and we need to better understand how we'll know when we're stimulating the correct area within that structure. Programming, I think there was a list shown going from Parkinson's through essential tremor. One of the things that goes along with that list is the newer treatments for OCD, depression, epilepsy. The programming is becoming more and more sophisticated or complicated, and that is one of the push for biomarkers, which is something we have here. We do not expect an immediate improvement in these patients when we turn the stimulator on. So when we're in the clinic or in the clinical room, we don't expect the tremor to stop and we can say, okay, go home, you're good. We have to find something that we think will cause an evolution of improvement over the next 4 months. And finally, as I think is going to be talked about better than I can do in the next talk, we're looking at delivery refinement, because right now everything we're doing is open loop. We're just chronically upregulating that pathway rather than trying to do it in any type of pairing with intent or rehabilitation efforts directly. I think with time I'll go ahead and skip that. Whoops. And so to summarize, new interventions are certainly needed to further promote stroke rehab. In our study, deep brain stimulation of the dentate nucleus has been well tolerated in 6 patients. We do have reversible stimulation-related side effects like most DBS therapies do. There's some vertigo that we get from the anterior context, but those are reversible and easily programmed around. The early data that I skipped through has tended to corroborate our preclinical findings, ability to modulate cortical excitability, and treatment-related changes in motor maps. Our outcomes are obviously variable with modest to marked improvements, and certainly the caveat needs to be expressed that this is a non-controlled phase 1 study. Acknowledgements for Andre Machado, my partner in this work for about 15 years, and the holder of this investigator-initiated IDE, as well as the large team that is needed to carry this out. Thank you.
We've talked about networks, targets, and applications, and now we're going to talk about mode of delivery. Steve Goetz of Medtronic. Thank you, Steve.
Thank you, and thanking the organizers and Dr. Morrell for the invitation. So I'll try to pick up a couple of the themes from earlier and talk about a journey, certainly towards adaptive therapies in this space. I'll speak specifically about movement disorders. Adaptive therapies exist in other aspects, epilepsy specifically, already. So I'll highlight movement disorders as an example of maybe more general progress that can be made. A few disclosures. I'm, of course, an employee, stockholder, patent holder, and I will talk about technologies, use of technologies that are in investigational phase and not currently available for commercialization. I start a bit with current state, and this was, I think, well discussed yesterday, but a number of points with therapy as it exists today. We do identify our indication very broadly based on clinical observations or responses. I won't read that indication statement to you. It looks like I got a little bit cut off, but levodopa responsive motor complications for a certain duration. These are observations in the clinic. We treat what is a very broad cohort of patients with very one-size-fits-all sort of uniform therapy. We deliver static waveforms, high frequency, somewhat titrated for patient-specific lead location, but it's generally a uniform therapy, statically delivered. And for this, we get good but not perfect outcomes. Generally, we treat certain motor symptoms, but not all motor symptoms. We increase the on-time for patients, but we don't treat all symptoms. We certainly don't treat non-motor symptoms. So I think this is a nice problem statement. We have a broad cohort that includes a lot of different symptoms within Parkinson's, including akinetic versus tremor, including other motor symptoms like gait, like freezing, like postural instability, as well as non-motor. We treat what is a very fluctuating disease state. We had some very nice talks yesterday about the on-off challenges with Parkinson's patients. We treat that with a uniform static therapy. And we get inconsistent benefit, and that it's inconsistent across time. The on-off fluctuations are clearly that, as well as differential response across patients and across symptoms. So the question is, can we do better? And I think we can. One of the strategies here is to use information in the brain to more tailor therapies for these disorders. And so as a bit of background, we understand that the firing patterns in neurons code information. Example of STN neuronal firing patterns here on our left. This has been used historically for targeting, for verification of targets. You see a typical trajectory moving down towards the subthalamic nucleus, one of the traditional targets for DBS, showing different firing patterns as you approach the target, within the target, and then after the target into substantia nigra. So this is useful information today, but we're increasingly understanding that these patterns are disrupted in disease states in predictable ways, and that's the insight that we hope to leverage into improved therapies in the future. So for moving from acute recordings in the OR, single unit, towards things that are more robust and reliable long-term, ensemble activity can be recorded as local field potentials. And I've got an example of these on the right. Information exists here, not in specific neuron firing patterns, but in the time domain behavior of that waveform, as well as the power spectrum. And we understand that both of these kinds of information are correlated at least with interesting things about clinical state. So on the right, we've got a early publication in this space from Peter Brown, who's now at Oxford, showing characteristic differences between an off state in a patient, off levodopa, Parkinsonian, rigid akinetic, showing a very, in both time domain and frequency domain on the bottom, very characteristic abnormal synchrony, let's call it, in beta. When that patient is on, you see a very different higher frequency signal in the gamma, and we're beginning to understand correlations of that abnormal synchrony in beta as a characteristic of rest or akinetic state, and those higher frequencies, gamma signals, as being signatures of prokinetic or motion. And as a final point, we can now make these recordings not just acutely. This 2005 publication done perioperatively, leads externalized, clearly not something we can translate out of the OR. These recordings can be made chronically, and one example of this is a research device from Medtronic, Activa PC Plus S, but I want to emphasize that other devices, including the NeuroPace RNS device, are available to do this chronically in other disorders like epilepsy. So it's becoming technologically feasible to get chronic insight into these networks over time. Now, to take that understanding a bit further, we're understanding that these local coherences are themselves indicative of network coherences. And so some of the very nice points made earlier by Dr. Rolston hold here as well. And I'm showing work here, 2 publications from Phil Starr's group at UCSF, showing on the left that the local coherence in STN is actually indicative of network coherence. So STN cortex, in this case M1 coupling, that is heightened, very present in Parkinsonian patients, not in dystonic or epileptic patients. Of course, we don't have access often to healthy brains, so we're not exactly sure what healthy brains look like, but we have a different movement disorder here and a non-movement disorder like epilepsy showing very different characteristics. So we can understand that this coupling, this abnormal synchrony, is likely a characteristic of Parkinson's, and given other work, likely a characteristic of the akinetic rigid symptoms of Parkinson's. So taking that itself a step further, on the right side here, looking at beginning to use this understanding to explore how DBS may work. And there are some references early to the initial ideas that DBS is a lesion or lesion-like effect. And what we're understanding is that it's more likely a desynchronizing effect. So when we see there's a laser pointer, oops, that is back. Laser pointer. Here is that abnormal synchrony phase amplitude before DBS, significantly disrupted during DBS, and interestingly, somewhat disrupted after DBS, even though often the clinical symptoms return quickly. Continuing this thought, again using some work from Phil Starr, summarizing an emerging understanding of signals of interest in this space. Again, sort of a schematic representation across cohorts of patients. Hopefully no individual patient has all of these symptoms at one time, but understanding that dystonic symptoms have a certain characteristic biomarker, tremor its own, that bradykinesia, rigid akinetic symptoms of Parkinson's in the beta band, and then dyskinesia in a very narrow band in gamma. So we're understanding these local synchronies exist, but importantly, in most cases, we understand these local coherences to be indicative of these abnormal network states. So one node in the network driving that network into a state that it can't get out of. So looking a bit towards a future state here, if instead of indicating around a broad cohort of Parkinsonian patients, perhaps we can narrow, given these understandings, our cohort to those patients with a specific network pathology. So in this case, the example I've given, excessive STN cortical coherence. If we then understand the disease state in that more narrow sense, we can start to target our interventions towards that network pathology. So choosing a waveform, a stimulating intervention, a control node, as discussed earlier, specifically tailored to disrupt that synchrony. Now, today that's high-frequency DBS, but it's very likely that with this understanding we can find either waveforms, frequencies of interest, or patterns, maybe on a population basis, maybe on a patient-specific basis, that disrupt that synchrony more effectively, more energy effectively than we do today. And then finally, because we know disease states change over time with medication intake, with sleep-wake cycles, with patient mental state, with motion intent, it seems likely that we should be adapting these interventions over time to the state of that network at a point in time. And I'll give one example of doing that, and that example is actually triggering the stimulation, this desynchronizing stimulation, to the beta bursts that we think we want to desynchronize. And as a final point here, it's very likely that in the fullness of time, this isn't a single intervention. We understand that patients have multiple symptoms. They may need a layered approach of control policies to address those symptoms, and we know states change over time. And so what is the best policy at a point in time may not be the best policy in that same network at a different point in time. So I think we need to face into this problem of control policy switching, or perhaps hierarchical control policies, dealing, of course, with the problems of interaction between those as we go forward here. But I'll start with a final example, or end with a final example, taking what is a fairly simple concept, but very promising improvement in intervention in this space. And so one observation in this space, and this is from Peter Brown's work again at Oxford, is that when we see abnormal synchrony in beta, it's not a uniform phenomenon. It's not the level of beta coming up uniformly. It's actually a bursting behavior. And you can see that in this picture on the left. These blue bursts, or in the envelope down here, are a representation of that beta. And in fact, it turns out that these longer bursts are the ones that are selectively or differentially driving the high amplitude. So there's a correlation between burst duration and the amplitude of those bursts. So if we think, we believe we have a correlation between high beta and disease impairment, we can explore that, and it actually turns out that those high Those long duration beta bursts here above about 500 milliseconds are positively correlated with clinical impairment, while the short bursts are negatively correlated. So here we're beginning to tease this apart. So it's not that beta is bad, it's that there is abnormal behavior in this beta region, excessive coherence, sometimes, that is driving impairment. So we're beginning to separate what might be, let's say, disease state or pathophysiology from normal signaling in this network. So if we carry that hypothesis one step forward, we may choose to deliver stimulation in a way that disrupts that pathology and allows the, let's say, normal communication, the normal coherence in that network to proceed unimpeded. And a very simple strategy here is simply to trigger stimulation on those long bursts with the goal of truncating or perhaps even filtering good behavior from bad. As a comparison, continuous stimulation here is basically suppressing everything, so the good with the bad. So we're down-regulating that network, which is generally helpful in terms of reducing symptoms of this disease, but it's probably also negatively impacting normal physiology, normal behavior, and driving side effects. And of course, the same group has explored all of these factors, and I'll emphasize this is very early work. This is primarily perioperative, meaning, again, leads externalized. This is an odd period for patients, just undergone a surgery. There are short-term effects called microlesion stun effects that do impact the symptoms in this time period, and these are small sample sizes. But in these populations, in this center's hands, they've shown improved motor performance. So this is that adaptive triggering stimulation on beta bursts, improved over continuous and certainly improved over a randomized sort of sham condition. They've also shown that that stimulation can adapt for changes in medication state. So here I have medication taken at time 0, wearing on, I should say, at about 40 minutes. You see the stimulation on time backs away from 50% into something like 25% in these patients. Interesting aside, these are two hemispheres from the same patient, and they are correlated but not synchronous. So there is a differential activity across the hemispheres, which makes sense as you understand this disease state, sometimes presenting unilaterally before it presents bilaterally. And finally, this group has explored side effects, specifically speech intelligibility, which is a known side effect of stimulation. So that idea of indiscriminate desynchronization of this network may not be great. What instead we might want to do is selectively desynchronize the abnormal physiology and allow normal signaling to occur, more or less unimpeded. So early work, but very promising. So I'll conclude here with a little bit of a vision towards the future. If instead of cohorting or indicating around broad clinically observable classes like Parkinsonian patients, if we can instead narrow down to a specific network pathology via continuous in-network monitoring or potentially non-invasive monitoring prior to implant, we should be able to get a much more precise cohort and potentially start to decouple symptoms from these broad clinical observations. So rather than treating Parkinson's, we treat tremor, and that tremor may present similarly across different indications. I think this gets very interesting when we start to look at, let's say, the more nonspecific non-motor symptoms of some of these diseases, like depression is a good example. Clearly shows up in psychiatry, but it shows up as a comorbidity in Parkinson's and epilepsy and post-stroke everywhere. And if we can understand the network pathology specific to something like depression, perhaps we can treat it for itself rather than as a collection of one symptom among many in these other disease indications. Similarly, if we can understand a little bit of the network pathology we're trying to get after, we can design a toolbox for that network pathology. So rather than one-size-fits-all high-frequency stimulation, we can look for waveforms that are really good at decreasing coherence, perhaps that are good at reinforcing coherence that's missing, and then that are targeted selectively at disease pathology versus normal signaling. And finally, we know these indications, all the interesting indications anyways, are dynamic over time, certainly in response to other interventions like medication, but also sleep-wake, also patient state, and sometimes down to the level of motor intention. So we know that beta changes when patients intend to move. And so to use that, we need to be able to adapt to that in a very short period of time. So certainly a little bit of a vision towards a future state, a lot of things that we don't know how to do yet, but I think starting to think, believe that starting to think about these sort of network state driven definitions will allow us to target more effectively, to design interventions more effectively, and ultimately to treat patients better. So with that, I'll close and thank, again, the organizers for the opportunity and everyone for their attention. And I think I hand off, well, I'll hand off right here.
Our next speaker will be Dr. Carlos Peña, who is Director of Rehabilitative and Neurological Devices. And I really want to say, I think you're getting a sense that this is a renaissance in neuromodulation, and none of this would be possible without a true willingness to, on the part of Dr. Peña's group, to support the efforts in developing and delivering safe and effective therapies. Dr. Peña?
Good morning. I always get nervous when I'm before lunch with a regulatory presentation. My name is Carlos Peña. I'm the director for the review division of neurological and physical medicine devices. And I'd like to thank the meeting organizers for inviting devices here to the conference, because it's so important that when you have a leading edge of innovation and technology, you have a portal to get those technologies to the marketplace and ultimately to patients. So I'm going to be reviewing a couple things, especially for the new people in the audience that I haven't met before. I'm going to be going over FDA review, a couple points to consider for neurostimulation products, and a few best practices and closing remarks. So here is our vision statement, and our vision is patients in the U.S. have access to high-quality, safe, and effective medical devices of public health importance and first in the world. And we take that pretty seriously. I have some data to back it up. Really, when I am in meetings, I'd like to look at it as a collaboration. It's not like FDA versus sponsors or companies. It's more of, we're both here to bring products that are safe and effective to patients. The definition of a medical device is that, there's a lot of definitions there. Instrument, apparatus, machine. What it comes down to is that if it diagnoses, it prevents or it treats a disease in humans, it may be a medical device. And so we begin to ask additional questions. It also works through its primary intended purpose through other means than chemical action, because then we would be down the drug pathway. And you can have a medical device even if you don't claim it's a medical device, and that's very interesting in the post-market space, which I'll be talking to as well. Here is our general approach for the oversight of medical devices. It's a risk-based approach. So we have 3 types of classes, class 1, 2, and 3. Class 1's low risks. They are generally exempt from our review. Class 2 are moderate or controlled risks. They require what's called a 510K submission. Typically, there's a product on the market, and if a sponsor comes in and wants to do something new with that product, we have some information about it. It can go through a 510K pathway. It's kind of like the ANDA pathways in drugs. And then there's the class 3 products. This is a high-risk classification. Requires what's called a PMA, or pre-market approval. And we provide oversight across these classes with both general and special controls, and where appropriate and necessary, clinical data. I'll talk about that in a little bit too. Here is a little bit more detail on the pathways. Class 3, generally PMA, pre-market approval. We receive several dozen PMAs each year, and these include our deep brain stimulation devices, spinal cord stimulation devices. Class 2, the pre-market notification pathway, we receive several thousand products, submissions in this pathway, and they can include some of the lower risk stimulators. And then there's the de novo pathway. devices that aren't comparable enough to something on the market. They're low to moderate risk. We haven't seen it before. We will evaluate that submission, and if we can get that to market, that'll serve as the predicate to subsequent products that are like it through the 510(k) pathway. I'll talk a little bit more about this. And then there's a humanitarian device exemption, regulatory pathway for products intended for disease or conditions that affect small, rare populations. These are generally small sample size studies, at least 30 patients typically, and it's a bar that it is for probable benefit but safety. I'll be talking less about that pathway. So the next question you might have is, when is clinical data needed, especially in an innovation-intensive conference like the one we're all at today? And so for PMAs, it's typically needed. For de novos, sometimes it's needed, but not always. And for 510Ks, it's not typically needed, although we've asked for clinical data in 510K. And so, you're probably saying, Carlos, really, when is clinical data needed? And the best way to find out about this is through a pre-submission. That's a way to get free feedback from the agency about what the clinical data requirements may be for your product. We love to have pre-submissions before you do the study, because then it shares with you our expectations, and we find out a little bit more about your product. To do even the animal studies, the non-clinical studies, to do the clinical study in the absence of coming to talk with us, especially on a high-risk device, is a little bit of a gamble. It would be ideal if we can sort of come to an agreement at the front end, rather than have to struggle with a data set that may not be the questions that we want to have answered at the end of this study, at the end of your development phase. There are typically 3 types of IDEs that we look at when a significant risk device is before the agency. There are early feasibility studies, less than 10 patients, small number of subjects, early in development. Then there's the traditional feasibility study. It's a larger study, between 10 and 30 patients. And then there's the pivotal study. That's the study that we look to make a marketing decision, typically. And that's appropriately sized based upon the questions that we're trying to answer, in generally based upon the preliminary data you've gotten in the early feasibility phases of your development. Feasibility studies are like phase 1, 2 in drug studies, and pivotal studies is kind of like the phase 2, phase 3 of drug studies. Please don't ask me to talk about IDE studies. I can't talk about IDE studies. They're proprietary, they're company confidential. I will be in a much smaller room than we all are here today if I talk about them. But this is where we take a look at those, we take a close look at what you are doing during a clinical study to get the data that we need to make a marketing decision. And it's called an IDE, an Investigational Device Exemption Regulatory Submission. And I'm going to talk a little bit more about this, but it's in the Code of Federal Regulations. It permits the use of an investigational device in a human trial. And here's some data. So this relates back to the vision statement that I had. Back in FY11, took us a long time to get a study up and running, full approval, more than 400 days. And this is a favorite slide of mine. I think some of you have seen it already. But FY13, we reduced by half. 15, 16, 17 fiscal years, we got it down to median time. That's a median time of 30 days. FY18 data is still being churned out, but I think it's tracking very closely to that 30-day median. That's like FDA's research data in a publication. That's like our Nature article. Those times are taken very seriously, because the faster we can get those studies up and running, the faster you can collect the data. Faster you collect the data, faster we can review it, faster we could potentially get your product to the marketplace. The only way we're able to do that is getting invitations from Dr. Morrell and others at conferences to share with you the expectations that we have. If investigators know what we're expecting with regard to safety, effectiveness, they're able to get their studies up and running very quickly. If there's a safety concern, those studies won't get up and running. If there's an informed consent issue, those will be disapproved. we take the safety, the informed consent of those studies very seriously, such that we can, stop those studies. But if you've submitted a free pre-submission, right, it's free, then you're typically going to know what we're looking for in your IDE study. That study is going to get up and running very quickly, and potentially we can have a research and development phase that's, your venture capitalists can get behind, because we're trying to make the regulatory landscape as predictable as possible. We're not making it less rigorous, but if we all have our expectations on the table, we're making it very transparent. So it's a calculated risk of developing a product, more so than just a calculated risk of like, oh my God, I have to deal with FDA. So a couple things about that process. Early collaboration is important. Use the pre-submission pathway, get feedback early. You can even schedule those meetings as informational, because then you can get an early look at what we're looking for. And this goes to sort of this next panel that, we are trying to move products to patients, to the marketplace, that are safe and effective. So there's a lot of panels shown here on different neurological and physical medicine devices, not meant to share with you the data sets that were involved. I mean, the first panel on the left was neurothrombectomy device. More than 100 subjects were in that study. Microcatheters on the right panel, there was no human studies involved. Those were 510(k) submissions. Purpose here is to share with you that each of these reviews was tailored to the particular risks and benefits, the safety and effectiveness of that product. Only way we can get to that understanding, to communicating those issues to physicians and to patients, is to having a good, robust relationship at the front end during the development phase. So a couple points to consider. One is on clinical testing, there's typically 2 parts in the device world. There's an exploratory part and there's a validation part. If you jump to the validation part, there may be some questions we have on whether we are really doing the right pivotal study, especially if the data comes out kind of unexpected. So we like the feasibility, the pivotal development, where appropriate. On trial design, especially for novel therapies, unlike the drug world, we look at what's called valid scientific evidence to make a judgment for a marketing decision. And we are required by law to look at a variety of different data sets, historical controlled data sets, single-arm data, randomized controlled studies, placebo-controlled. There is a variety of trial designs to choose from based upon the device, based upon the patient population, based upon the indications for use, based upon what we know about the device, based upon what's done overseas. So, you kind of it gets very tailored on what the right study is. A randomized control study may be the right study in some cases for devices. An open label design may be appropriate for another type of device. So for example, it would be very hard to have a randomized sham controlled blinded study of a prosthetic arm. Like, how do you blind that? So what's important here is that we choose the right study design under a pre-submission, and we choose the trial design that will have the least amount of uncertainty for us to make a decision. It's never about, sometimes I hear stories about, oh my God, we have to go to FDA and it's going to take so long, FDA. It's never about the regulatory process. It's always about the scientific or clinical considerations and trying to evaluate what the safety and effectiveness is about a product. Those are not regulatory issues. Those are clinical, scientific issues that we need your help with in determining. Neurostimulation, couple additional things. Safety. We look at all adverse events reporting during the clinical study. We're interested in short and long-term safety issues. Acute, usually within the first 3 months, depending upon the product. Long-term safety data. Typically, we may ask for 12-month safety data. And we want to know what happens with the device. Could work out great for the first 3 months, but then, if little issues come up, that's important to know. And usually, we would have had some signal in the feasibility portion of the study from which to design a really good pivotal study to help address and mitigate those risks that we might have seen in the feasibility portion. Effectiveness is also important. So it's reasonable assurance of safety and effectiveness. It's very comparable to the drugs world. We want to make sure medical devices are safe and effective. So endpoints need to be meaningful, validated. There are times when we look at unvalidated measures, but those need to be in, we should have a discussion about that with criteria, with the time of assessments, with an understanding of the variability. What's the inter-rater variability of that assessment that you're doing to get outcome data? What is the patient variability of that measure on effectiveness? Does it vary over time? And does that variance, is it the same magnitude as the difference you got between the treatment and the sham group? That's an important question. And a few additional points to consider. Typically, a single pivotal study is what's needed to make a decision. That usually works out well when we have a good feasibility study from which to base and depart our measured assumptions. Trials for medical devices can be difficult to blind, so we would want to have potentially a blinding assessment at the end of the study. And recommend evaluating your device in the population indicated, but at the end of this study, how much can you generalize the results? Because that makes, if it could help a broader population with the information that you have collected in the feasibility and pivotal study, that would be great if we can generalize those results. And when is that appropriate to do? And then the endpoint should be pre-specified for safety and effectiveness. We don't like post hoc analyses. It should be pre-specified to help us make sure that what we're studying is potentially a true effect with the least amount of uncertainty. Just a couple more slides. Organization, we're sort of relatively new, a couple of years. Division of Neuro and Physical Medicine Devices. It's one of the newest. It is the newest division if you compare it to others. We're in the Office of Device Evaluation. There are a number of other offices. That yellow row is within the Center for Devices. There's a research component also. There's a compliance component which looks at post-market activities. And then there's a surveillance and biometrics. When a product goes out into the marketplace, it's usually based upon a pivotal study, but that pivotal study is not going to be anywhere near the population that's using that product. So there may be signals that we need to track during its post-market use. Interestingly, we are now moving towards a sort of a new structure, a reorganization at Center for Devices, where we're going to be combining the Office of Device Evaluation, the Office of Compliance, and the Office of Surveillance and Biometrics into one office called the Office of Neuro and Physical Medicine. We're really excited about this new structure because now sponsors can come in, talk to one team that has the pre-market side of review and also the post-market side of concerns that would need to be addressed. So, they're getting ideally the sponsor is getting a really broad look at what the regulatory landscape looks pre-market and post-market from one team. And I think that's helping also institutional knowledge, institutional care of the products that we work with during their lifespan. Currently, though, we have five teams: diagnostics, interventional, 2 neurostimulation branches, one in neurology, one in psychiatry. It's kind of like a fuzzy border, 'cause sometimes they cross one another, but we have 2 branches that are involved in this area. And pre-submissions, this is the best way to get feedback from the agency, especially prior to an IDE or marketing submission. You can even submit a pre-sub if you have a non-significant risk device, because if you're wanting to market that product, potentially, if you get some early feedback on the design of that study, even if it's a non-significant risk, it could be very helpful to the types of questions that we want to make sure are answered prior to a marketing decision. And it's also free. So a couple final closing remarks. One is, engage early. We get that it takes a lot of money to develop a medical device. We get that non-clinical studies include a price tag, and the clinical studies include a very big price tag. But if you engage early, you get a little bit more information about what that regulatory landscape looks like, that could help make business decisions, hiring decisions, financial decisions that I think you would want to know early on. And it may be just good information. It may not be that bad, but we should have that discussion early on. Two, and if I haven't mentioned it before yet, pre-submissions are free. Use them. I mean, they're free right now. It's no excuse not to come to the agency to talk about your product that you want to get to a patient. And three, if you've been thinking about like the dry cleaning today, or what am I supposed to do for grocery shopping, there's my contact information. So there should be no reason why, if you have questions about contacting the FDA, you have someone to talk to. I think you'd be surprised about the interaction you might get at the agency. Thank you.
Thank you so much, and if I could have the speakers here. I think that what you've heard is that unlike some medications, we can personalize devices in real time, and they're modifiable, and unlike some ablative or surgical procedures, they are reversible. So these are all great advantages and one appeal to devices. Let me open it up to any questions. Please.
A question for Dr. Peña. Could you talk a little bit about the breakthrough program for devices and how that's going?
Sure. So we have a breakthrough program for devices that it used to be called the Early Access Program, the EAP program, where if it's addressing a debilitating disease, or it's an improvement, an enhancement to an existing product that really shifts the treatment and the care of patients, we will review those submissions with additional resources as we can, with the limited resources that we have, and try and expedite that product to the marketplace. So it usually involves additional staffing assigned to that submission once the EAP designation is given. And we try to also clarify at the front end the development plan, lay out the regulatory requirements in a little bit more detail than we typically do for the general submissions. For PMA applications, it's probably a lot easier to do because those have a little bit longer timelines. For 510Ks, which are within 90 days, we may be able to shave off, a week off the process if it's an EAP designation for 510Ks. A week off the 510K process, I mean, I don't know what you all do, but that's a big deal for FDA to shave off a week. We're trying to improve our numbers. The way we improve our numbers is having the same expectations that you all have with early interactions, which we can make the EAP process succeed.
Okay. Hi. I was interested when the guy in the plum shirt talked about, I think you talked about, some others maybe did too, about the where you discovered that things became different, and then you had to, after you had the study going, and then you had to go back to FDA to get permission to adjust what you were doing. And so my question is, how does the FDA react to that kind of thing? Because it seems to me you're in the middle of a study, and now you gotta, change it. And how does that work? Or does it? I think you said it. I may be wrong.
Yes, I'm the one that brought it up. How the FDA reacted, I can't honestly say, since Andre holds the IDE.
I can't hear you. I'm sorry.
Sorry. Any better? Okay. So yes, I was the one that brought it up. It was the 4 to 8 month change, and essentially there are. I could get myself in trouble with this because I don't actually do the IDE part. I leave that to Dr. Machado. But we looked at that patient, and you have windows to notify the FDA. You have windows to, we have contacts that we go back to at the FDA. And it really comes back to, Andre is the holder of this IDE. The patients are his responsibility, and what goes on is ultimately, when it's sort of one of those where he's looking at a patient that's continuing to get better, and he has to make the decision of continuing the therapy and going back to the FDA to try to get that approval. And the timeline of that can be a little bit, is where we get in trouble, because we might not notice that until it's about the time to turn it off. So this gets back to the speed of those communications. Again, I can't really specify what happened in this case, but essentially we changed the protocol. We went back to the FDA to change the protocol. They were very responsive and approved the change.
So, that was my concern, because you just, you got to keep going here.
Yes.
for the patient, and and the FDA is open to, and pretty quick.
Yeah, definitely. I mean, especially during the early feasibility and the traditional feasibility study phases, if there are changes, especially with regard to safety, if there's a way to improve the safety of a trial, or they want to add a measure, or they want to modify the products with software updates, there's a number of different submissions that one can submit to FDA to make sure that we continue to roll with that study, but those changes are implemented, so that when we get to the pivotal study, things are a little bit more locked down. But during the early year of feasibility, traditional feasibility study, you can sort of make changes, and as long as patient protections, patient safety is in place, we're willing to work with folks.
Okay, because that was a very interesting thing to me, that things change as you're doing your work. And
Thank you. Ken?
just another
Oh,
interesting thing about our brains.
Ken?
Yeah, hi, this is for anyone on the panel, probably mainly Dr. Peña, but anybody can jump in. I've worked with oral medications for years with the FDA, very fruitful, very happy to do it. But in every project, we always wanted to know the ceiling. And I'm wondering with I guess electricity is sort of like your dose here. Do you have to take it up to a certain level that would tell you it's sort of toxic, or you don't want to go there, and so you know what your ceiling is? And does that vary from patient to patient?
Yeah, I'm certainly happy to answer that one. Yeah, so it's there's some interesting pictures earlier of the volume of tissue activated. So we have fairly good stimulation models of the spread of that stimulation, and what happens depending on where that lead is targeted. The stimulation will, at certain levels be within the target of interest and be within that network, and at some point will spread outside. And typically that spreading outside, at minimum it's wasted energy, but often that drives side effects that can be observed. And so you'll start to activate other networks that have deleterious effects, be they motor, be they other side effects, paresthesias. In DBS specifically, you're near internal capsule, which can have impacts elsewhere in the motor system, dysarthrias, double vision. There's a number of side effects that are easily observable when you start to deliver too much energy because you're spreading outside of the target of interest into other targets.
Do you find that really varies from patient to patient, or can you pretty much know
Yeah.
where you're going to get into trouble?
So it's the big variable in DBS specifically, but in a lot of neural interventions, is the accuracy of the lead placement. So surgeons do the best job they can do. They use all the tools they can to verify that location, but there's still errors of, in the published literature, 2 millimeters. That sounds like a really small number, but some of these structures are no bigger than 3 by 5 by 4 millimeters. So you start to look at those error stack-ups, and you might be in the middle of your target, you might be on one side, you might be on the other side. And so one of the major variables patient to patient is lead location relative to anatomy. There are also likely patient-specific factors like excitability of those networks, like the size and shape of their anatomy, like other pathologies they might have that might limit the impacts. So I think there are patient factors, and then there's this big factor of how close did you get to your desired target, knowing that you're probably not perfect.
Great. Thank you all very much.
Yeah, it was very interesting to me because I'd been involved as a site investigator in a number of pharma trials that we figure out that we titrate the dose to some extent in each individual, which is quite different. I do want to ask, I have a burning question, because this was the other thing that has struck me is it has to do with trial design. So we look at the standard pharma trials. And I assumed when I came into this field that we would use what is done pharmaceutically. And maybe, John, you can answer this first. We are dealing with what we recognize now to be an implant effect of uncertain duration. There are acute effects of stimulation, but there are also clearly chronic effects that may take years to appreciate. So might you just comment on how you think about that in terms of the duration of a trial? And then, Carlos, I would really like to think how your division thinks about trial designs, understanding that we're dealing with multiple mechanisms of action with different timelines.
That's a very good question. So when we do, for instance, epilepsy stimulation, we implant the device, and then we will sometimes see an sort of microlesion effect, especially with the anterior nucleus of the thalamus, where seizures are helped immediately, and then they recur over weeks or months. And then there's the programming part of it, which can take again weeks to months to get right, which is a long process to know exactly what parameters are best for an individual patient, and they are individualized. And then there's that long-term, what we think is some sort of neuroplasticity, which is engendered by the chronic stimulation. And that can be over the course of years. And we see that in the long-term follow-up data where we look at 3-5 years out, we see improving outcomes, which we don't believe is all just due to loss of patients or loss of follow-up. So how we should be separating all this out, for the long-term stuff, I don't know, because it gets to be infeasible to do a study for 10 years to know if the device works, even though you'd love to have that data. So we have to, I think it'd be great if we could have some biomarkers like we had alluded to before that were more rapid and we were reliable to know the device is working. We could speed up programming and get rid of some of that slowdown. But it is an unsolved problem currently, although people are working on it.
How is the agency thinking about this?
Yeah. So, we don't have a group of elders in the basement that we go to for all our answers. We like to have early feasibility studies try and characterize what outcomes we might receive with a particular product. So, baseline phases are important. Post-surgery phases are important. Programming phases are important. The acute phase is important, the chronic phase is important. So if you can sort of characterize different phases and understand what each phase has the impact upon the treatment of a particular patient, that's going to be key. The other thing to note is that there may be different disorders that wrap around neurostimulation a little bit differently. Like, I think there's some early indications about dystonia being a little bit more longer responsive to DBS versus other movement disorders. So, there's also a knowledge, intellectual, clinical component that we have to sort of fold in. And then the third piece of information is we sort of scour the literature for those very, single case study publications to make sure that we try and pull apart as much information as possible from those initial physician experiences. So if you can get the trial, under pre-submission, if you can get the trial well delineated in the early feasibility stage, a little bit about the disorder, about the sort of use of it, hopefully in the U.S. early, first in the U.S., then you could sort of lay out a very well-controlled, reasonably controlled pivotal study from which to make information available to clinicians to use with their patients.
Bob?
Really great session. Thanks so much. I think my question is for you, Dr. Baker. When you have the electrodes into the dentate nucleus and you then repeat the rehabilitation prior to starting the stimulation, are you able to record any activity from the afferent system, fronto-ponto-alveolo-cerebellar system, that might circle back to dentate? And if so, would that information be any help in predicting who might respond?
Yeah, so the two slides that I skipped are essentially relate a lot to the electrophysiology opportunities that are built into this study. In the OR, we try to, although the patient's anesthetized, we do somatosensory evoked potentials. We do what I'll call deep brain stimulation evoked potentials, where we actually stimulate each contact at given parameters and look at the spatial temporal mapping of that across corkscrew EEG electrodes implanted. And then actually from there, like they talked about with the Brown work, we do externalize the patients for 7 days, meaning we actually have access to the 8 contacts of that implanted lead for 7 days after implant and for 7 days just prior to explant. So we are even able to look at a pre-post therapy type of situation. Across that 7 days, we do as many different things as we can, looking at coherence between the dentate and scalp EEG, essentially in this case, at rest, during motor tasks. We do cognitive event-related potentials, eye tracking tasks, because the dentate's is known to be involved in saccades. So we are looking at quite a number of different metrics, partly as biomarkers, partly for basic interest, etc.
Well, I think that you've heard today that there's some pretty extraordinary fundamental science going on in this field, but at the same time, we're actually delivering therapies to patients. So please let me know if there are particular topics you would like to hear in the area of neuromodulation next year, because we're going to make sure that we maintain a strong presence at this meeting. I want to thank our speakers so very much. I think we got a good overview of where we are and how we are all working together as a community. Tom Satula asked me to remind everybody that after our networking lunch, we will be breaking into 2 sessions. So you'll look and see there's 2 opportunities, and you can select the one that you wish. You'll probably be split. You'll probably have to send half of yourself to one and half to the other. And thank you all for your attention.